Hello students, our next topic that we're going to be talking about with Evolution 101 is mutations. So before we get into mutations themselves, we need to do a little bit of a genetics primer. So I like to think of genetics being sort of like a recipe book. So if we're working with this analogy, the chromosome would act like the whole book itself. If we zoom in a little bit to the gene, a gene would be like the directions for making a specific type of cookie. And then if we zoom in even further, we're looking at this picture down to the DNA level, the order of the bases codes for specific ingredients for the cookies, or in biology land, amino acids. The amino acids are what get mixed together to actually form a protein, or if we think of our analogy here, the cookie. And then all of these proteins or cookies come together to make up our dessert tray, which contributes to the meal. So why am I telling you all of the different levels of baking and genetics here? Well, when we look at what goes into making traits of an organism, this is really the flow of what happens. So the DNA, which codes for the protein, helps to determine the characteristics of the cells and the tissues in a particular area, and all of the cells and tissues come up to make a structure or a trait. So the traits, in other words, the traits in an organism are determined by development of cells into tissues, which is controlled by DNA and the proteins that they code for. So if we're talking about mutations, mutations are a change in the DNA. It's a really simple, direct explanation or description. So if you look here, we've highlighted a base. Over here, we have like a chunk of a chromosome. And so this is a direct change to the DNA itself. So if you think of how this fits into our flow chart, if you have a change in the DNA over here with DNA and protein, it could change the cells and the tissues that they make up. And if you change the cells and tissues, it could likely change to the traits and structures that they develop into. So an example would be like limbs. So there's a lot of different causes of mutation, but the most common one is really copy error. So copy error being like if you ran something through a copy machine and it came out, but it wasn't perfectly clear. So your cells make copies of the DNA every time that they divide. So this picture over here, or what we call a fork, is showing that you have the old DNA or the original DNA, which is blue, and then you make a copy of it. Okay, so you get a copy. And they should be identical because you're making them off of the original strand. But a mutation would be just basically a mistake. So somewhere that when the enzyme was putting in the bases, it put in the wrong one. Often our cells will correct this, but if it doesn't, it can be a mutation that could potentially be passed on. Some of the other causes of mutations are more external. So one of the most common ones that we can think of is UV radiation. So that could be either going out in the sun or going into a tanning bed, which you should never do. X-rays and things like that are more minor for what we do, but if you work with them all the time, it can be a risk factor. A lot of these are choice related, like cigarette smoking. So you can choose not to do them, or benzoyl peroxide, looking at your ingredients. So there's a way to avoid excess mutations, but they are just a factor of biology. So we want to talk a little bit about the types of mutations so that we can understand why everything doesn't change every generation. So the first type is substitution. So this is a change in a single base. So you can see over here, we have CTG, GAG, and the change, if there's a mutation here, replaces one of the A's with a G. So this can result in a few things. So a missense mutation would be a change in amino acid, so one of the things that makes up a protein, because of this mutation, which could potentially affect the protein. A nonsense mutation means that this would turn into a stop signal, so your protein might be truncated or too small, and that again could affect the protein's function. Or it could be silent. So there's some redundancy in how the DNA code actually codes for the amino acids that go into the proteins. So it could be that this base doesn't really make a larger change outside of just this base pair. Some of the other types include, get my picture out of the way here, insertions and deletions. So this is pretty much what they sound like. You have an increase or decrease in the number of bases in that given area. So if you see here, you have this six letter code, but we've added 3 more in. Or down here, you've taken 2 out. So if this is in a region that actually codes for the amino acids that make the proteins, it could be that now you're missing pieces or you have extra pieces, and that can again affect the protein's structure, which would affect its function. The last type of mutation that we'll talk about is frame shift. So this changes how the sequence is read. So we read DNA when it goes to be made into protein. The directions come in sets of 3. So for example, over here, the fat, cat, sat. So these are all 3-letter words here, and they all mean something different, kind of like our amino acids. If I take out the T though, and I still start with the first one available, I still have to read in groups of 3. So I have HEF, ATC, ATS, and then this short AT that may do nothing. So basically it's shifted the frame that you're reading, and that could have a very significant impact on what amino acids go into making the proteins. So one of the things we talk about with all mutations is randomness. So we might be able to predict that mutations will occur. So there's a certain rate that they will always be present because of just copy error in the DNA or just external influence from being alive out in the world, but we can't necessarily predict where they will occur in the genome and what their impact will be. So for those reasons, we consider mutation to be a random mechanism of evolution. We also always talk about variability when we're discussing mechanisms, and in this case, mutations will always increase variability. And this is because they can create new genetic information, which means that there's basically new stuff in a population. So if you look at the picture here, this is looking at how you have a sequence that through reproduction, so sperm and egg coming together, and development through embryo will give a particular phenotype. But if you have a mutation in that sequence and go through the same process, you may get a different phenotype. So this means you've basically made new traits or new phenotypes that exist. And so for that reason, we consider mutation to be something that increases variability in a population. So to put it all together, remember that at its basis, mutation is a change in the DNA, and a change in the DNA can lead to a change in traits or structures. It is random and it increases variability.